Hello everyone, this is River and I'm a PhD student of University of Maryland, and my project is about bioaccumulation of DDT and soil remediation. So we know that DDT is an organic pesticide and it is also persistent, so it can accumulate it in the body of the animals, so that for the contaminated site, DDT will be concentrated through the food chain, and finally the concentration gets very high in the animals like the birds or mammals, so which is bad. In order to protect these animals, we need to decrease the exposure of DDT to these animals. Traditionally, we have like physical, chemical, and bioremediation, but these methods are very expensive, which is not feasible for the sites like this, for the very large site. So we need to find an alternative method which is inexpensive, and traditionally, and the previous study finds that the compost like this is able to decrease the DDT bioaccumulation in the lab or in the greenhouse, but the effectiveness of this compost in the field has not been investigated yet. So for my study for this project, we are going to investigate the effectiveness of the compost to decrease the bioaccumulation of DDT in the field condition. Here is a map. So this is the map of the project. Oh, the light is very bad. So basically, we have 7 plots on the sites, and for each site, we have 4 different treatments, and one is control, another one is till, and another 2 are 2 kinds of compost. So here is the site. So the site is this. So we can see that this is one plot, and it's very hard to see the boundaries, but we can see it's very grassy. Okay. So basically we have 7 plots in the field, and in each plot we have 4 treatments, and for each treatment, and then we are going to sample, and then we take soil and earthworm samples before the treatment, and then we get samples after applying the treatment, and then we can compare the effectiveness before and after the treatment. And now I'm going to show how I do the sample. Because this site is very grassy, I'm not going to do in the real plots, I'm just going to do it near it. So we are going to get soil and earthworm samples on this site. So basically we dig the soils and separate the soil apart, and then we can look for the earthworms like this. And then we can collect the earthworms in a jar. And then we save the soils in the sieve like that, and then we have the saved soil, and we collect the saved soil. So this is soil on the same side. So finally, we are having the earthworm samples, which is not enough for one analyze, but I just show how I do that. And here's the soil sample. So finally, we just take soil and earthworm samples to the lab to analyze the concentration of DDT in these samples. And that's it. Thank you.